This mini lecture is going to discuss a process called DNA replication. Our key concept is that DNA replication copies the genetic information or the DNA of a cell. So for DNA replication to happen, a single strand of DNA, half of it, serves as a template for a new strand. And then the base pairing rules will direct how the replication happens. So remember, our base pairing rules are A bonds with T, C bonds with G. Now you guys have already learned that DNA is replicated or copied during the S stage of the cell cycle, synthesis. And then finally, at the end of the cell cycle, each body cell gets a complete set of identical DNA because an exact copy was already made. So let's look a little further into how this process actually works. So remember, we kind of talked about how the DNA will serve as a template. Well, then the actual work is done by these specialized proteins called enzymes. So the first thing that these enzymes will do is they will unzip the double helix DNA. They will separate it into the two sides, two equal halves. Next, free-floating nucleotides will come up and they will form hydrogen bonds with the template strand. So you may remember a nucleotide is made up of a phosphate group, a sugar, and one of 4 nitrogen bases. Now those nitrogen bases, thanks to base pairing rules, always try to pair with the same thing, A with T, C with G. They find each other and they form one of those weak hydrogen bonds that we've learned about. And that helps hold them in place. Next, this very special enzyme called DNA polymerase comes through and it actually bonds the nucleotides together and starts forming that double helix that we know of. Polymerase enzymes are what actually form those really strong covalent bonds in the phosphate sugar backbone. Finally, we're left with 2 molecules of DNA, and each has an original strand and a newly formed strand. The way that we talk about this is that we say DNA replication is semi-conservative, or DNA is semi-conservative. And what that means is that for each molecule of DNA, it's going to have one original strand that it saves or conserves, and one brand new strand that was just formed. Finally, this process of replication is really fast and really accurate. So in organisms like us that have a lot of DNA that's really long, replication will actually start at multiple points in our DNA. And you notice going from A all the way down to D is that as replication happens, it gets wider and wider and wider in all these points until we're left with the 2 different strands of DNA. Something else that's kind of cool, this enzyme's DNA polymerase can find and correct errors. So every once in a while, the base pairing rules don't work. A might accidentally pair with C, G might accidentally pair with T, something like that. This enzyme DNA polymerase actually detects that, and it will break that weak hydrogen bond, and it will kind of usher in this better nucleotide, the one that's supposed to go with it, and it will fix the error. That is all for this lecture. Please let me know if you have any questions.